episode 2, and we are going to be talking about the passengers of the ocean. Now, the ocean has these huge currents, it's doing all kinds of things, and it's moving, those ocean currents are moving huge amounts of water around the globe. And it's not just water that they're moving, because if you're small, and not, especially if you're not a very strong swimmer, you're just going to get carried along wherever those ocean currents are going to take you. But they're important, the things that are carried by those currents, because that means you can potentially be moving huge amounts of nutrients or life around the globe, or heat. And so the passengers of the ocean are really important, and that's what we're going to be talking about in this episode. So to start us off, we'll begin with the passenger of the ocean that is probably most famous at the moment for a really sad reason, and that is plastic pollution. So let's have a look at how it moves around the ocean engine.
You can think about it this way. So it's like if every minute a garbage truck's worth of plastic is being dumped into the ocean, which is a staggering amount. The plastic, once it enters the ocean, it moves around. It's affected by ocean currents, and marine animals can also interact with it and move it to different places. And eventually it accumulates in different compartments of the ocean, like the ocean floor. Some sink to the ocean floor, some accumulate on beaches, some are ingested by animals. So larger plastic objects, they can lead to entanglement of animals, and they can also be ingested, but the smaller plastics are more likely to be ingested because of their size. So animals eat particles that are smaller than their gape size or their mouth. So because of the small size of microplastics, they have a greater ability to interact with food webs. As a result, they can be passed through food webs and accumulate in top predators. That's kind of one hypothesis that people are exploring. And they also contain chemicals that can leach into the animals. So it's both physical impacts and chemical impacts. We know that production has been increasing. There was a study done in 2017 that estimated an 8.4% increase in production, plastic production every year. We're not sure how that translates into increase in environmental concentrations, but a recent study done in the North Atlantic did find an increase in surface concentration of plastics over time. So we know that the concentrations are also increasing, parallel to the production. I also study climate change, and there's this global kind of target for climate change, right? The 1.5 degrees Celsius number. Unfortunately, such a target does not exist for plastic. And that's something that has been the topic of discussion of a lot of people. What's the tipping point? How can you come up with a tipping point like this? And it's something that does not exist at this moment. A lot of the attention, especially at the beginning of the field, focused on charismatic megafauna. So animals that are cute, that are lovable, right? But right now, it really has become an issue that is human-centered. And we know that there are a lot of impacts to the laboratory organisms that people have tested. And therefore, it makes sense that it would translate into human health as well, impacts on human health. Of course, it's unethical to dose a human with plastic. So that has not been done directly, but we can infer from the other studies that there are indeed impacts to humans. And like you said, it is becoming more of a human health-centered problem nowadays. It's really important to reach out to our political representatives and authorities. And that is definitely a very effective method to get the word out, is to interact with the people who are supposed to be representing you and making a difference. And it's very easy to feel depressed or alone in the situation. Like when you see your cucumber wrapped in plastic, you're like, what do I do about this? But it's important to stay positive. I find that taking action really helps to combat that kind of anxiety.
Some of the most underappreciated passengers in the ocean are life. They can be single cells, tiny little things, so bacteria or algae or fungi, and the ocean is full of these tiny passengers, but they are far too small for us to see with our own eyes, so you need the help of a microscope. And those can be really expensive and difficult to use, but there are some clever people working on this. And so next we went to meet an organisation that is trying to make microscopes and the ability to see into this tiny world of life accessible to everybody in the world. And that is Fairscope.
I'm a postdoc in Minute Prakash's laboratory at Stanford University. And so that lab is interested in a lot of things, but most of all making frugal science tools. So making tools kind of democratized so more people can use them than just scientists.
Cool.
And
And,
so,
and this is one of them in front of us here.
yeah, this is one of them. This is the plankton scope. So this is a frugal instrument to look at what's in the water, so notably plankton, as you can see behind me here. And so it was made to kind of bring plankton imaging beyond just scientists that maybe can't afford, a $100,000 instrument. Because a large problem in looking at the ocean is it's just very big. So it's hard to sample it with a few very expensive instruments.
So let's look at this. So this is actually, it's made of wood on the outside here, which is really, I love that.
Yeah, so we wanted to also make it more sustainable than a lot of the other instruments. So it's made out of recycled bamboo. And so it's very delightful to machine while also being very robust. So the other great thing about this is you don't need to be worried about taking it to the field. So I even just throw this in my backpack. You can power it with a battery and then take it on a kayak wherever you're looking at plankton. Especially for people that say it's very important to look at plankton. So people that have a lot of these harmful algal blooms, a lot of them can't afford the very expensive instruments, and they're also in places that maybe don't have internet connectivity or something like that. And so for this, you don't need connection to any internet. You don't need special software. So just from your cell phone, you can use this and start taking images right away.
So let's look at the bits here, because it looks a little bit complicated, but tell me so presumably there's some water in this tube here. Is that your sample?
So this is where you load your sample. This is set up kind of for lab use, but you can imagine you could just put this in a puddle or in the ocean if you want to just pump right from there. So the sample flows through here. It's quite simple. This is just a little bubbler to keep our stuff happy. And yeah, it's just a pump. So it's pulling the sample through here. And then this is the flow cell that it goes through.
So it's one of these bits of tube. So the sample is flowing through the tube, and then you've got a microscope that's looking through that hole at what's in the tube.
Exactly. So there's just a light in front that's illuminating. And in here is the camera, and there's a lens system here. And then there's a Raspberry Pi inside that is controlling everything.
So these are all quite cheap components, basically.
Exactly. And it's also, like, the Raspberry Pi community is wonderful. It's like another nonprofit organization that is like very open and used by a lot of people that are making these frugal instruments, while still being a very powerful and reliable computer. So
just before we get to what you can see in the water, let's just deal with the concept of small things living in the water, because when you scoop up a handful of seawater, it doesn't look like there's anything there, right? But there are passengers in there.
Yeah, exactly. And a lot of people, when you look at the ocean, you can kind of abstract it as being this mass that can just absorb everything that's happening, but is actually filled with all these creatures, which are actually, notably they're sequestering, anywhere from 40 to 60% of the carbon dioxide that we're emitting, through the carbon emissions. And we really don't know what's happening with them. Most likely, they're being negatively affected by a lot of the human impacts. So we just don't even know the state of plankton as it stands. And so just visualizing them, I think, really helps people, not just scientists, but kind of respect the ocean as a living system.
Because they are amazing, aren't they? The thing about plankton is there's sort of no rules almost. I mean, there are, because they have to swim in water and it's quite viscous for them, but they've got little legs sticking out and antennae that do all kinds of different things, and they're just amazing different shapes. It's quite an astonishing world, I always find, looking at the plankton.
Yeah, and I mean, even just as objects, they're interesting, right? So I think, even going back to like art and design, you can find really wonderful things just looking at the shapes. So, I mean, even I just look at these. I am a scientist, but I just appreciate them for like the beauty that you can see. And there's always something new wherever we've gone with the plankton scope. For instance, in New Orleans, we went to just a little river and we took a sample, and there's just things I'd never seen before inside there. So it's always a surprise.
So the ocean is, I think that's one of the wonderful things about the ocean, that the passengers it's carrying, they might be invisible, with our own eyes, but they're being carried, they're part of the fabric of the ocean almost. They're being carried along with it, and they are the most important living part of the ocean.
Exactly, and they're very hard to see, so you don't normally think about it.
But this is bringing those passengers to an audience that wouldn't normally get to see them.
Exactly, so hopefully you can bring this to more people, not just scientists that will want to see what's in the water, because it is helping people study the ocean, but also just helping people respect the ocean and understand, that it's a living thing.
might sound a bit strange to say that some of the passengers of the ocean are fungi, but we're not talking about mushrooms or toadstools here. There are other types of fungi in the ocean, much, much smaller, and they may be very tiny passengers, but they're doing some really interesting things. We're right outside the poster session. There's like literally 1,000 or 2,000 scientific posters back there. And I'm here with Shui Feng, who goes by Nick, who is one of the scientists who's got a poster in there that caught our eye because you work on fungi. Now, what are fungi doing in the ocean?
We think they are degrading carbon that has been produced by phytoplankton and all the other primary producers as well as secondary producers. They might be parasites, they might be saprotrophs, and they might be pathogens, but we know very little about them, especially in the open ocean.
So there's different habitats within the ocean. So there's things that can be completely free floating, and then there's things that can stick to the edges, basically. And we normally think of, mushrooms in our everyday life, for example, they're kind of stuck to trees and things. They grow out of trees.
Yes.
So do you get those sorts of fungi in the ocean, or are they different sorts of fungi?
No, we don't get so much mushroom-forming fungi. Yeah, and in fact, people have ignored fungi in the ocean for a long time because they think there's just not enough substrate, not enough food for fungi to grow in the ocean. But from the molecular data we have, using both DNA and RNA sequence technology, we have found that there's a really wide diversity, an array of different fungi in the ocean. But we still are trying to have a grasp on what they're actually doing in the ocean.
And what do they look like then? So are you finding them just using eDNA, so environmental DNA, where you kind of scrape up everything that's alive, read all the DNA barcodes of everything, and then say there must be some fungi in there? Or are you actually finding some fungi and looking inside that?
We're doing both. So we're also using cultivation. So some of them look like yeast. Okay, so they're basically tiny microscopic cells, about 2 to 10 micrometers in size. And some of them are filamentous. So those are the ones that can develop these root-like structures like rhizoids and physically puncture into large particles and perhaps break it down for the larger microbial community and for them to have access and use it as food for the entire community.
So there's an important point here about how dynamic life in the ocean is, isn't there? That normally when we think of life on land, we think of kind of big things that are slow. So, a tree or a cow, and they're very large, and you definitely know where they are, and they live quite a long time. But in the ocean, we have this thing where things are very, very small, and they live very, very quickly, and they don't really store stuff. So it's, a tree is loads of stored life, right? Whereas these small things in the ocean it's just the life, and then they just keep living and dying really, really quickly.
Some do, and some are just slow growing because they might be found in the bottom of the ocean in the sediments where the turnover time of all organic matter is very slow. So they might just subsist on a very low amount of organic carbon, and they might live for longer than just a few days or a few weeks. And it's still yet for us to find out how long they could live.
So give me an example of one, fungi have species, right? I know they're a bit weird.
Right.
So one species of fungi, and tell us what it does on it. How does it start? Where does it live?
There is this phylum of fungi called chytridiomycetes, and they are best known for their role as diatom parasites. So diatom is one of the most primary producers in the ocean, and they account for almost a 1/5 of the oxygen we're breathing in and out right now.
So when we're talking about primary producers, we're talking about the harvesters. They're harvesting sunlight, basically, and then other things can eat them. So they're the equivalent of plants on land, that they collect the energy.
Exactly.
Everything else can use it.
And they form the basis of all food web in the ocean.
And we're talking about single cells. Diatoms are single cells.
Yeah, they're small, typically less than 100 micrometer in size.
They're really tiny. Yeah, so something that small has a parasite.
Yes, they have a parasite. And what's amazing about diatoms is that they have these shells called frustules made of silica, just like glass shells. And fungi actually evolved a mechanism to use their chitinous cell wall and punch through that glass cell wall. And then they could suck up the juice in there, eventually develop and mature into a rhizoid. And once they mature, they would harbor over hundreds of little spores, zoospores we call them, on the inside. Eventually they will burst out and swim around and try to encounter another diatom cells again.
Why are the fungi different, or why do they matter?
So they are important in the sense that they're important in terrestrial environments where they play a very important role in recycling the carbon that have been fixed by the primary producers, such as trees and grasses. So in the ocean, they are also very important in recycling the carbon that have been made in the surface ocean where there is light, and return the mineralized form of nutrients back to the rest of the ocean.
And just finally, I mean, this conference has so many big things at it. Like, so we're only 3 days in as we speak, and my head is already about to explode.
Likewise.
But I just wondered if there was something in a different field that really stood out to you, or something, you know, something you'd seen that you were really like either impressed by or shocked by, or that was really interesting. Is there something that comes to mind that you saw and you were like, that's really cool, or maybe people don't know about that?
Well, I was at this plenary session yesterday given by Dr. Don Canfield, and he was talking about these sponges, which is one of the earliest form of animals on Earth, go through these contractions, and when they contract, they actually goes completely anoxic. So it's an animal, so it requires oxygen for them to breathe. And the main function of sponge is to filter seawater. But why do they go anoxic? Is it just a form of an adaptation?
So basically they stop breathing for a bit.
They stop breathing for multiple hours every day, and they have a cycle of about 12 to 15 hours.
Wow, I didn't. Is that all sponges, or is that just some of them?
It's one of them. So these sponges produce spicules, and these are not the glass sponges that would be more difficult for them to contract.
That's great. So, the sponge, which is an animal, I always think of sponges as barely counting as an animal, because it's basically a lot of stuff with a little bit of animal kind of woven through it, just enough to count.
Right.
But so they're sitting there on the seafloor, they're filtering water, and then they shrink and stop breathing for a few hours, and then they kind of grow back and just keep going again.
Right.
That's bonkers.
I know. The theory is that maybe they're exposed to too much oxygen, so they're trying to stay away from the oxygen.
Hide.
And just repair themselves for a few hours.
That's amazing. I love the way that you walk around there, and you know, a lot of it's sort of sensible, sort of the sort of science you'd expect, and just every so often there's something like that.
I know. He had this amazing video of the sponge contracting. So.
Oh well, I'm sorry I missed that now. We've heard about the problem, the plastic, and how it's moving around the ocean. But the next question is, what can we do about it? So next, we're going to hear about some interesting and innovative ways that might help us deal with the plastics problem.
We are dealing with PHB, polyhydroxybutyrate. It's a class of PHA. And what's exciting, especially exciting about PHB, is that it can be degraded in light-deprived, cold, marine environments, which a lot of the bioplastics on the market, that's just not viable. They're only compostable in an anaerobic digester or something with high heat, really specific microbes in there. So
this is the idea that these things that people say are biodegradable plastics often aren't biodegradable, because you have to put them in very specific conditions before they'll break down.
Exactly. It's a policy thing. You can kind of say it's biodegradable if it's in this really specific environment, but as we know, a lot of plastics end up in the ocean. It's not like an apple where if it's in the ocean, it will break down eventually. It really depends on the microbiome.
Well, let's start at the beginning then. So why would a bacteria be making a biopolymer or a bioplastic? Like, you know, they're busy, they're getting on with living, they're taking in nutrients, their metabolism is turning over. What's in it for them making bioplastics?
So it's not that they're necessarily making a bioplastic, it's that they're making a polymer that they can store the way we'd store fat. So the PHB is stored in a granule in the bacteria to be used for energy. So we are turning it into a bioplastic. We are seeing the capabilities of that PHB and adding additives or whatever we need to do to make it viable as a plastic alternative.
So potentially there's a material here that you can make something out of, and then let's say it's a plastic cup and it ends up back in the ocean somewhere. How is it definitely going to biodegrade? Does it need to be near the right kind of bacteria? How hard is that next stage?
It does need to be near the right kind of bacteria, and that's definitely closer to my research. This study that we did was looking at the ecological diversity. What kind of bacteria is able to degrade this bioplastic, and then in addition sort of looking at really specific genes that are interesting from an industry perspective. And I guess I can mention this here, that one sort of further down the line goal that we have is to be able to incorporate a bio-printed sticker. So this is actually being developed at Ann Meyer's lab at the University of Rochester. It's a gel sticker that could be incorporated with the PHB product that's being deployed to time the degradation. So you have the PHB product and then you have the sticker that has bacteria, viable bacteria embedded in that, incorporated in the ocean to time that degradation for a certain time.
So basically it has a little factory with it to break it down when the right time comes. That's amazing.
That's the ideal plan, and that's way down the line, and there's lots of obviously lots of things involved in that to really time the degradation in a usable scale and an industrial scale, but that's the goal.
I love that idea, just to dwell on that for a minute, that you can create a product which is carrying with it the things it needs to degrade back into the environment, and that then therefore wherever it goes, when the time, I guess the sticker, it's got some timer, so it releases them, and then it's time, and then they just eat it.
Yes.
And that's it.
That's the goal.
That's amazing. And so just tell us a little bit about the progress towards that goal. Where is it now?
Absolutely. So we are, on my end, we're able to isolate more species of bacteria that we've found to have this capability. We're sort of looking at each of those isolates and deciding, based on certain genomic capabilities, which of them are viable for that kind of use in like a sticker or something like that. So one really important thing is looking at PHB depolymerase, so the genetic capability, even if we saw in the lab that it could break down PHB, the genetic capability, confirmation, this isolate can degrade PHB. Another sort of side of that is looking at, we wanted to look at the presence of alginate lyase, and we wanted to sort of see, alginate's a really common carbon source in the ocean because it's coming from brown kelp. And the thinking is if this bacteria that we're using is potentially able to use alginate and PHB, it may preferentially use alginate, and actually also those bio stickers are alginate based.
So alginate is something else that algae produces, another type of goo, basically. The ocean is full of goo.
Yeah, right. It's a really common carbon source, so that's something a lot of marine bacteria are able to break down. And ideally, the thinking is, okay, we would prefer to use something that only can break down PHB and not alginate,
right?
So that it doesn't just break down the sticker and whatever else is around. We can really time and control the degradation.
So we'll only eat one thing,
yes,
basically.
the thing we wanted to eat. And that's, I think, also the biggest challenge with a lot of microbial, bio-based alternatives is there's a lot of timing involved. There's lots of things you have to look at that you may not consider just on a surface level.
And so, I mean, I know these are impossible questions, but if you were going to pick like some number of years ahead when we might actually be able to buy something that was made of this, like are we looking 10 years, 20 years, further than that?
That is a great question. So right now, Mango Materials is a fully functioning company. They are making these products, PHB products, that are available, purchasable by customers. And then on the flip side, having something incorporated in the degradation, it's hard to say. There's definitely a lot more research involved, but I would hope in the next 5 to 10 years, ideally. My PI may look at this and be like, what are you talking about? But that would be the hope, but yeah. And
just talk to me a little bit finally about this idea that, you know, there's loads of amazing things in the ocean, and basically a lot of life in the ocean is it's either fighting or eating or surviving. It's doing all these things, and they've evolved to suit all these niches, and they've got the genetics that lets them do these things. Just talk to me about the reservoir of potential in the ocean.
Wow, that's a huge question. I would say that I think the most exciting thing about biology in general and looking in the ocean is that there's so much to be discovered. There's so much that's existent in the genomic capabilities, as you say, of the bacteria that's there, of the animals that are there, that actually can be utilized to rethink how we approach these sustainability solutions. So the possibilities are endless.
Brilliant. Well, thank you very much. Good luck with your work. So that's it for our story on the passengers of the ocean, but we have one more in this series still to go, because next time we're going to be talking about the voyagers, so do join us for that. And so that's it for this one. If you like what we at Cosmic Shambles do, please do look at the links down here and like and subscribe to the Cosmic Shambles channel. And while we love doing this, we can't do it all for free. So if you like what we do, please do support us at patreon.com/Cosmic Shambles, because that helps us keep it freely available for everyone. And we really do want to share the good stuff as widely as possible. And so for now, from the Ocean Sciences meeting in New Orleans, goodbye.
Thank you for watching. I hope you've enjoyed it. Even if you haven't enjoyed it, I still think you should support us at patreon.com/Cosmic Shambles. So, and also, by the way, click and subscribe. That's what they say, don't they? I'm 55 and I'm having to say click and subscribe. I mean, just for the loss of dignity, you should click and subscribe.